Okay, so hello everyone. My name is Beth. I'm a fourth and final year PhD student at the University of Aberdeen in Scotland, and I'm very excited to be here to present one of my PhD study chapters, which is non-linear transcriptomic responses to compounded niche transitions across temperature and resources in a pest beetle. So many species around the world have or are currently experiencing drastic changes to their environments due to anthropogenic events. And these changes can be changes in abiotic factors such as global climatic changes and/or changes in biotic factors such as changes in resource availability through land use change. And some species are able to survive the changing environments through plastic and adaptive mechanisms. For instance, they could expand their geographic range or become resource generalists. But it's still important to study the general processes that are governing these responses in order to better understand how species are rapidly responding and surviving to changing environments. And most studies that aim to predict responses to environmental change in species manipulate only a single dimension of the environment, so just changes in resources or just changes in temperature. And while that is important for studying the direct effects of a given stressor while controlling for confounding variables, different components of the environment rarely change in isolation, with many circumstances leading to organisms experiencing simultaneous changes to multiple aspects of the environment. So for example, if a species is expanding its geographic range in response to temperature changes, it could subsequently experience changes in resources, as was observed with the brown Argus butterfly in the UK. So with heterogeneity increasing with environmental change, it's key to study the combined effects of multiple aspects of environmental change on the physiology and fitness that may enable individuals to rapidly respond and survive to changing environments. And these responses to multidimensional environmental change can be additive or non-additive responses. Additive responses occur when the effects of multidimensional environmental change is the sum of the effects to single-dimensional change. So if temperature and resource change causes a change in response, this can lead to similar changes in response to simultaneous temperature and resource change. Non-additive responses are when the multidimensional environmental change causes a response that's different to the response under single-dimensional change. And this can be split into synergistic responses, where the response to multidimensional environmental change is greater than the response to single-dimensional change. And antagonistic responses are when the response to multidimensional environmental change is less than the response to single-dimensional change. So it's important to study the multidimensional interactions in order to understand the context dependence of environmental change on physiological and fitness responses, because if we don't, then we may assume that some responses occur across all contexts. And I'm quite interested in understanding the responses to environmental change in insects, as many are capable of rapidly adapting to environmental change, particularly insect pests that are increasing their geographic range in response to changes in temperature and resources facilitated by things like the increase in global crop trade. And so studies on the molecular and phenotypic responses to multidimensional changes would therefore allow comparative approaches to better understand insect adaptation, as well as aiming to improve pest management in agriculture. And I used the seed beetle as our model system for our study. So Callosobruchus maculatus has been increasing its geographic range to new climate regimes globally, alongside its ability to rapidly colonize a wide range of beans. So this study had collected beetles from different subregions around the world, and they were found to be colonizing different bean types. So for instance, the beetles from Africa and South America were found on cowpea, whereas the beetles from Asia were found on beans like adzuki and mung beans. So this shows that the seed beetle is a global cosmopolitan pest species that can rapidly colonize new resources and thermal regimes, making it a good model system for our study. So the aim for our project was to study the transcriptomic and fitness responses to single and multidimensional environmental change in insects. We predicted that the fitness responses to single dimensional change would be exacerbated by multidimensional change. And we also predicted that the multidimensional changes would interactively impact gene expression by creating non-additive responses to multidimensional environmental change. And our overall goal was to better understand the molecular and phenotypic responses that may enable insects to rapidly respond to multiple aspects of environmental change. So our experimental design consisted of 4 treatment groups: the control treatment, which was the ancestral bean of cowpea on the ambient temperature of 27 degrees, 2 treatments that represented single dimensional changes, so the first one being resource change of either chickpea or common pea novel bean at the ambient temperature of 27 degrees, and then the elevated temperature of 35 degrees on the ancestral cowpea, and finally the multidimensional resource and temperature treatment, so either chickpea or common pea novel resource plus the elevated temperature of 35 degrees. The beetles were kept on one of these 4 conditions for 2 generations, and then I measured fitness traits on the second generation, which was the F1 fecundity, F2 developmental period, and F2 offspring viability. I also collected larvae from the second generation and measured gene expression responses using RNA sequencing. And for those treatments that had resource change, I could only get larvae from the chickpea for RNA sequencing. We used DSEC2 to look at pairwise comparison differential expression analysis, and EDGE-R to specifically look at the non-additive interaction effects of temperature and resource change on gene expression. So when looking at the effects of single and multidimensional environmental change on our fitness traits studied here, we found that resource change in chickpea and common pea, which are the orange and green lines shown here, had an overall negative effect on fitness by decreasing fecundity and offspring viability while increasing developmental period in comparison to the control treatment, which is cowpea 27, which was the blue line. The single dimensional change in temperature had little overall effect on the fitness by reducing, they only reduced developmental period. And that's quite common in insects in response to elevated temperatures. But when looking at the multidimensional resource and temperature treatment, we found that it had a significant interaction effect on fitness by creating larger fitness differentials than in either resource or temperature treatment alone. In other words, the combined resource and temperature treatment had a greater effect on fitness than either resource or temperature change did alone. Our between-group principal component analysis shows that the variation in gene expression among the treatment groups is best explained by temperature change. So we can see here that the resource group of chickpea 27 degrees groups quite closely with the control group. So this implies that the among resource variation in gene expression is lower in larvae experiencing ambient temperatures of 27 degrees compared to those experiencing elevated temperatures or the multidimensional resource and temperature treatment, as these groups are further away from the control. So this analysis provides initial indication that elevated temperatures exacerbate transcriptomic responses to novel resources. Our Venn diagram here shows that the single dimensional change in temperature caused the greatest number of differentially expressed genes compared to the control at 1,314 differentially expressed genes, followed by the multidimensional resource and temperature treatment, and then the resource treatment alone caused the least number of differentially expressed genes compared to the control at 222 differentially expressed genes. The smaller numbers and the colors associating them within each circle represent the number of up in red and down in blue regulated genes within each treatment, and then the overlapping circles represent the number of shared up and down regulated genes between treatments. So we actually found that relatively few genes were shared between treatments. So for example, only 308 genes that were upregulated in the temperature treatment were also upregulated in the multidimensional treatment. So this implies that a different set of genes is differentially expressed in the multidimensional treatment, suggesting that single dimensional changes do not cause the same transcriptomic responses as multidimensional changes. Our EDGE-R analysis to specifically look at the non-additive interaction effects of resource and temperature change on gene expression found 115 genes responding non-additively to multidimensional resource and temperature change. So that means that these genes responded differently to multidimensional resource and temperature change than they did under each condition alone. We categorized these genes into 50 specific functions, and grouped them based on 5 common functional trends. But in this talk, I'm just talking about one particular trend where the majority of these genes, so 69 out of 115 genes, were overall upregulated in the multidimensional resource and temperature treatment of chickpea 35 degrees. And these genes had several protective mechanisms, and I highlight a couple here that I think are quite interesting. So the endopeptidase cathepsin L was upregulated, and we know that it's expressed specifically in the guts of insects and is involved in protein degradation. But it was also found to be upregulated in thermally stressed psyllids and stink bugs that were fed a generalized diet. So this implies its importance in responding to resource and temperature change. So our findings agree with our predictions that fitness responses would be exacerbated by multidimensional changes, as the multidimensional resource and temperature treatment cause larger fitness differentials than in either resource or temperature treatment alone. And we also predicted that the multidimensional environmental change would interactively impact gene expression. And we did indeed find 115 genes responding non-additively to multidimensional environmental change. So just to summarize, we found non-linear or non-additive transcriptomic responses to multidimensional environmental change in resource and temperature. And these genes identified were involved in several protective and repair functions. We conclude that the single-dimensional change alone cannot determine fitness and transcriptomic responses to multidimensional environmental change, meaning we need to study species experiencing multidimensional change in order to understand how they are actually responding to a heterogeneous environment. And finally, we suggest doing some validation analysis on the genes with these protective mechanisms and looking at how they respond to multidimensional environmental change in the long term to assess whether and how these responses may be evolving over time. And with that, I would like to thank my co-authors, Leslie and Aoife, and thank you to my funding body and to Novogene for helping out with the RNA sequencing. If you have any questions, my email and Twitter account are on this last slide, so just feel free to send me a message. Thank you.